Hello friends, I am Bhujbal Pratiksha. So today I am going to talk about DNA replication. So the DNA replication is the process by which cell makes the identical copies of DNA. So this process is important for the cell division, growth, repair, and the transmission of genetic information from one generation to the next generation. So the for the DNA replication there are several steps. So first one is the unwinding. The process with the unwinding of double stranded DNA molecules, it the enzyme called helicase break the hydrogen bonds between the complementary base pairs, that is adenine with thymine and cytosine with guanine, which separating the DNA strands. And second one is the DNA template. Each separated strand serves as a template for the synthesis of new complementary strand, and these templates are referred to as a parent or template strand. Then third one is the complementary base pairing. Then free nucleotide in the cell's nucleus, which are the building blocks of DNA, are used to create a new strand, that is adenine pairs with the thymine and cytosine pairs with the guanine. These complementary base pairing ensure that the new DNA strand is an exact copy of the original. Then fourth one is the DNA polymerase. So the enzyme called as an DNA polymerase synthesis the new strand by adding a nucleotides in the one by one to the growing chain. Then the DNA polymerase can only add a new nucleotide in the 5' prime to 3' end direction. So it synthesis the new strand in continuous manner along one of the template strand. Then fifth one is the Okazaki fragments. For the other template strand, that is lagging strand, DNA polymerase synthesize short fragments in the 5' to 3' direction away from the replication fork. These short fragments are called as Okazaki fragments. Then sixth one is the RNA primers. Before DNA polymerase can start synthesizing a new strand, a short RNA primer is added to provide a starting point for DNA polymerase. So the RNA primer is the enzyme which is responsible for the synthesizing these RNA primers. Then seventh one is the DNA ligase. After both the leading and lagging strand are synthesized, these are the gaps between the Okazaki fragments on the lagging strand. An enzyme called as a DNA ligase joins the fragments together to creating a continuous new strand. Then proofreading and error correction. DNA polymerase also has a proofreading function to ensure the accuracy during the replication. If there is any incorrect base pair is added, it will be removed and replaced with the correct one. Then termination in the replication is continue until the entire DNA molecule is copied. Once a replication is complete, there are two identical DNA molecules, each consisting of one original strand and one newly synthesized strand. Then these molecules can be separated and distributed to daughter cells during cell division. Thank you.